This is a slide of a cat eyeball. Here is the slide of the cat eyeball using the 4x objective, and in this part of the slide we're actually looking at the retina and the choroid layer and the sclera. Here I've increased the magnification to the 10x objective, and again we can see the sclera, the choroid with the pigment, and the retina. And we can see a lot more detail in the retina. We'll label the parts of the retina at the next microscopic level. Here is the magnification using 40x objectives, and in this we can see a lot of different things. Here we can see the rods and cones layer. We can see the cell bodies and nuclei of the rods and cones. We can see the cell bodies and the bipolar neurons here, and we can see the ganglion cells along this band. Here is a closer view showing the choroid and the pigmented layer. There's the rods and cones, and in this slide they're actually separated. Now we're going to look at the cornea and the ciliary body of the cat eyeball. We're using the 4x objective here. Now we're using the 10x objective on the cornea, and we can see a little layer of stratified superficial cells. We can see this basal layer of cuboidal cells, and then all of the stroma of the cornea. Now I've increased magnification and I'm using the 40x objective. We can see the stratified cells and the basal cells. Here's the stroma. Now we're going to look at the ciliary body and the lens. Now in this slide, using the 4x objective, we can see several things. Here's the lens. These are ciliary processes. And there's the ciliary body. This would be the cornea. And that wraps up my discussion of the cat eyeball slide.